Agent Music, Special Agent 99, Maxwell Smart.
From Microbe TV, this is Q&A with A&V. I'm Vincent Racaniello, and joining me tonight from Chevy Chase, Maryland, Amy Rosenfeld. Hello, Amy.
Hello, Vincent. How are you today?
I am very well, and I have to tell everyone, Amy is having suburban freakout. Right?
Yeah. Oh, yeah. I've seen the inside of the car and Rockville Turnpike more than anybody ever should, especially within a 74-hour period of time.
Well, the good news is Amy went 3 times to Best Buy yesterday so she could get all the equipment she needed to do this live stream. And so we are the and you're in your new home there in Chevy Chase, right?
Yeah, I'm in my new home in Chevy Chase. And you left off, it wasn't just the 3 times to Best Buy. It was the 8.5 hours of on the phone with Comcast.
Well, it's working. It's done, right?
8.5 hours of Comcast. There's just so much hold
So
music one should be
Elena
subject
says,
to.
do you have a new cashmere wardrobe for Chevy Chase?
I will, for sure. Not yet.
Yeah. Anyway, isn't
I
that
will.
cool? From Chevy Chase, Maryland. So
And no, I'm not sitting in a closet.
what do you mean? No, it's
Somebody
dark
asked.
because
Kathleen P says I'm sitting in a closet, and I'm not sitting in a
no, she's
closet.
not in the closet. She's in her living room, but she only has a little reading light because we haven't set up her lights that our kind listener sent. They will be set
Richard,
up.
don't forget Richard.
Richard,
That's Richard.
we will set them up, and we will. Amy
Well,
hasn't
we're
found
moving.
she needs to move the desk and so forth, but she hasn't had a chance. So be kind to her lighting tonight. And I mean, she did this so that you guys could get your Amy fix. So yeah, she doesn't have a sweater tonight. No, you don't. You have some kind of a thingy. She said the
I
striped or
have
the
a
tangerine.
I have a white 3/4 sleeve shirt.
Okay.
Um, it's 92 or something. Hold a second. Got to push all these
It's
buttons
92
now.
degrees out.
In Friendship Heights, it's 88 degrees. It was 94 as a high.
So would you
Yes.
rather introduce you from Chevy Chase or Friendship Heights?
Friendship Heights, Chevy Chase is a
Chevy
neighborhood.
Chase.
I don't know.
Chevy Chase is good. I like the sound of Chevy Chase.
Yeah, me too,
Okay.
because I can see it on the sign on the highway when I'm speeding past the exit going,
So Amy
why did
does.
the GPS lady tell me?
I have to say, Amy didn't have a car in New York, of course, for the past 8 years, correct?
Oh,
Yes.
longer.
And so she had to buy a car to go down there, and she doesn't like driving. She hates it. She said, "I just want to
Yeah.
take the bus and the subway," so she needs to get used to it. So
Yeah.
you're there for your new job. Are you going to reveal it yet tonight or another time?
No, it's pretty much done. We can discuss it.
So Amy, where are you going to be working? Tell everyone.
So we've moved. The lab and me has moved. You notice how grammatically correct that is? The lab and me, not the lab and I. The lab and I have moved to the Center for Biological Evaluation and Review at the FDA
And
in
so
Silver
you're going to
Springs and
you're
White
going to
Oak,
have a
Maryland.
you're going to have a lab where you can do your enterovirus research, and you also have to do some regulatory business for FDA, correct?
Yes, it's like instead of writing grants, it's a
Okay,
trade-off.
folks, did you hear that? Amy is going to CBER of the FDA. It's the research arm for biologics, Center for Evaluation of Biologics and Research of the FDA. It's a great job for her because she doesn't need to write grants. They're going to pay for her research, right?
It's a great job. Not just only that, it's great colleagues and everything. It's great. It's not just about writing grants. It's
So,
about support and intellectual. It's about intellectual support.
so I just want all
And
the
there's a ton
listeners
of really smart people.
I just wanted the listeners to understand that Amy will be reviewing biologics that are submitted. So you're in good hands, folks, because you know she's a critical person. And biologics include what? Vaccines, monoclonals, that sort of thing?
Hmm.
Okay, good. We look forward to hearing. Well, we're not going to be able to hear it. You can't talk about this stuff, but when you publish papers, we'll hear about it, right?
Yeah, you'll discuss them on Twitter, I'm sure. I'm sure
So,
I'll tell
so
you.
Doreen wants
I
to know
just won't discuss
if you're
them.
going to have a side gig at a university.
I hope to be an adjunct professor at universities in the area. Yeah.
Yeah, that's always a good thing to do, so you can, you know. And so this, you folks are seeing Amy's career evolve here. As
Evolve.
Yeah,
Evolve.
evolution is a good thing. Can be a good thing, right?
Hmm.
So you've heard her talk frustratingly for the past few years about funding and research, and now she has her own gig, and you're going to see her blossom.
Yeah, that'll be good.
And
Now I get to hire people.
she
People
gets to
can
hire
send me
people.
resumes.
She's going to have several people working with her, right?
Hopefully.
Amy's going to be the boss.
Oh yeah. I'm a big mean boss. You should talk to Edmund and
And and
Audrey.
and listen, live stream colleagues and friends. She announced it here tonight. She hasn't told anyone except her family or a few other
Well,
people
that's questionable too.
and me. Okay. Well, we are very excited for you. Now, the frustrating thing is you cannot take the metro to the FDA, right? You have to drive.
Not easily. Not easily.
It's a shame. It's a shame. It would be perfect because the Friendship Heights metro is close to you, right, where you live?
Yeah, 7-minute walk.
Yeah, it's too bad. And if you took, what would be the closest metro to the CBER, to the FDA?
I think you take it to, I think, well, the closest is Silver Springs, but I think you can go to Bethesda, like outside the actual NIH
Hmm.
research part, and then you take a shuttle. And I don't know how long that shuttle takes, but it doesn't run frequently enough for me. It's not good timing.
Well, let me officially publicly say, Dr. Rosenfeld, I'm proud of your
Yeah,
accomplishments. I'm
it's
very
good.
proud of this position. I look forward to your developing and further development into an even more accomplished scientist. I'm sure you'll do great stuff.
Yeah, I'm excited. Thank you. I'm really excited. We're going to
Yeah.
be good. It's going to continue the legacy and stuff we built on and stuff that you built on that you forgot.
Yes. So my lab is done. My lab is done, and it moves. It moves. All the reagents move to.
They in the freezer and they get on truck and they come to Silver Springs. Yep.
All right. So Eleanor wants to know which of your virology lectures do your students like the most and the least? Oh, I can tell you. The most is to toss up between transformation and oncogenesis and therapeutic viruses. And the least, they don't like the structure lecture. It's too bad because I think it's fascinating. I love virus structure.
I
And
think they get confused about the triangulation number.
yeah, triangulation number is hard. Yeah. All right. Michael wants to know, was there a Paxlovid Epstein-Barr Association rebound that I heard of or that I dream that? Do you know anything about a Paxlovid EBV?
No. But I've been off the radar for a few days.
You drove from New York to Chevy Chase on, Monday, right?
Mm-hmm.
Memorial Day.
Mm-hmm.
Okay. Okay, Amy, how long does it take to set up a new lab? What's the best new feature? Do you know what your lab looks like? You've seen it, right?
I've seen the physical space. Yeah.
It's relatively new, correct?
Yeah. It's less than 10 years old. It's only 8 years old.
So how long will it
So
take you
I've
to set
already
it up?
set up a lab. So it should take about 3 weeks or less. I should be able to do some experiments within 3 weeks.
Yeah. And if you don't, I'll hear about it, right?
For sure.
See,
For
Amy,
sure.
Amy still has my email. She has my text number, my cell phone number. So there's no escaping her. right?
No, no. I'm like the octopus.
Your recent award medal some weeks back was fantastic. Kudos. Thank you very much. I'm glad
Great.
you liked it.
It's good.
And oh, did I help with packing up the lab for the move? Actually, Amy hasn't moved any of the lab stuff yet. We cleaned out the lab. I helped her over the past few weeks to throw a lot of stuff out. And much of what's left she wants to ship down there. And she thought it would be easier once she got there to direct that. And you're actually going to come up in a couple of weeks and pack everything up with me and ship it, right?
Yeah.
And that includes viruses, cells, reagents, plasticware.
Yeah. Antibodies, plasticware. Yeah.
All that
Medium.
stuff.
Enzymes.
We're going to give FedEx a lot of business.
Yeah, the FedEx man should just stand outside the lab and bring his little wheelie thingy.
Yeah.
Maybe he might need more than one wheelie thingy. You know, they called to ask me if we wanted the blue container removed from the lab. I said no.
No, it's not ready yet. Congratulations on your new position. We are all very happy for you.
Thank you.
Will you miss New York City restaurants?
Yeah. I don't go out to eat a lot, but yeah.
What's the next big virology conference? I guess
going to
it's
ASV.
The American Society for Virology, which is in Madison, Wisconsin in July. Now, next weekend, not this coming weekend, but the next weekend, the American Society for Microbiology is having its annual meeting in Washington. I'm going. However, there's not much virology there. It's mostly a bacteria/fungi kind of thing. I forgot to thank our moderators. Let's see. So far, I see Les. Thank you very much for being here. I just saw Vanity Nutrition. Who else is here? Let's scroll back up. Tom is here.
Steph?
I don't see
Where's
Steph.
Steph and Tom and
Tom
Frank?
is here. Tom is here. Les and Vanity Nutrition. So thank you all for
So
coming.
then where's Frank?
Tonight. Frank is not here either. Thank you all
There's
for
no
coming
Steph
again.
nor Frank?
No Steph nor Frank. But
We
that's why we have so many people, so that if a few can't make it,
I've
it's not
yet
a
to
problem.
see Animal Party either. No Animal Party.
No animal party. She'll probably pop in later. Yeah. So let's see. Get back to the bagel. Are you going to miss bagels? Philip says you're going to miss bagels. But you can find good bagels there. Isn't that place by the University of Maryland has good bagels? But that's a hotel, right? You can't buy bagels there.
You think I'm going to drive to the University of Maryland hotel to buy bagels? Seriously, you think I'm going to drive to get bagels?
There's probably a store in Chevy Chase.
I already looked it up.
Yeah, where is it?
I looked up the 10 best bagel spots.
Oh,
That store at the hotel was number 3. Then there's Bethesda
Okay.
Bagel, which is number 2. And oh my God, listen to this. There's a branch near me.
Excellent. So let me know how it is. All right, here's the first sciencey question. Will global warming cause the number of pandemics to increase?
Most likely.
So scientists do not predict. They just explain. It's my new mantra. But
Really? You have a new mantra? Did I approve this mantra?
no. I'm not going to follow it. It's a good idea, but I think what do you think, Amy? The effect of global warming is we did that paper. Oh, you were on the TWIV with Dixon. We did that paper which was a theoretical modeling of the effect of mass animal movement. And because,
due to global warming.
Yeah, global warming. So animals
where-
are going to move to their comfort place, and then they're going to take viruses with them. So it's very likely that will contribute to more pandemics. Yeah. Yeah, it's unfortunate. It's unfortunate. And so, yes, Amy's at the FDA, not Howard Hughes. And it's a great job, folks.
Yeah, I'm super excited.
From now on, whenever possible, if I'm public speaking, I hope there's a French horn quartet. Thank you. Yes, it was very nice. And I told you that they wrote, one of the French horn players wrote a piece for that ceremony, and he called it, Richard Ernst interlude or something, CD155, which is the poliovirus receptor, which was discovered in my laboratory a long time ago. So it was very nice of him to do that, right?
Mm-hmm. It was wonderful.
And do you have your Manolos there you could show people? Are they packed away?
Seriously?
I'm just curious. You didn't unpack them yet, right?
No, I barely unpacked. Let's see. I spent all day on Comcast phone and then at Best Buy's, where I'm now on good relationship with 4 of the 5 people to help me find anything in the store because it's disorganized, or at least I think it is. And then today I went to Home Depot, got lost. Got lost. Bought the wrong thing at the Container Store because now my silverware doesn't fit into the drawer. Not so good. And and I felt, and if I had other time, free time, I filled out 46 pages of forms.
You have to get clearance of some kind, right?
Yeah, but I'm not worried. I have clearance from the FBI. I'm not worried. FBI might be worried, but I'm not worried.
No, if that you needed that to work with select agents, right? If they work in the BSL-3.
Yeah, go to Ian's, yeah.
Suburban freakout. Okay. Is it possible to isolate a wild-type virus for viruses with a high mutation rate? Would each variant be the wild type? So
you don't know what is the variant and what is the wild type when you isolate something. So the first thing you isolate, you call is the wild type.
So wild type
the way it works.
Is the thing with wild type. It's basically what's in nature, right? And sometimes in the old days, you would get, say, poliovirus, wild type polio. You'd bring it in the lab and you say, this is wild type. And here's the vaccine strain that was made by Albert Sabin. But yes, if you isolate from many, many people, they're all variants of the wild type, right? That's how I look at it.
Well, there is no wild type.
It's kind of a reference.
Usually right. It's usually the first one that you observe, that you isolate. You call the wild type or the reference strain, and then you compare everything against it.
Exactly right. Here's good stuff on filtrable. The term applies to the filtrate or the residue. So both the virus and the bacteria are filtrable. Amy was right again. Hey, don't let it get to your head.
It's okay. It's not going to get to my head.
And this is the thing. Amy gets it done. Yeah. Yep. Welcome to Maryland. I guess Caroline lives in the Maryland, D.C. area.
Cool. She can be helpful since all I do is get lost. One day I'm just going to lose the car. I walked around the garage the other day trying to figure out where on G1 the car was. And then I walked past the car.
You get used to it.
Not so good.
Amy looks remarkably fresh for someone who just moved. Thank you, Amy.
That's good. Thank you.
Lots of congratulations. Thank you, Rob, for your contribution. Really appreciate it. This week in fungism. Ask not for whom the shroom extols. 'Tis all for the greater glory of fungus. My gosh, Rob, you have such creative fungal things.
Yeah.
What percentage of people who have taken Paxlovid get rebound? I think it's 1 to 2%, right?
Yeah, somewhere.
Which is about what was. Remember, this was seen during the clinical trial, but the press didn't make a big deal of it. So
they didn't even read the clinical trial report.
Yeah, that's right. I mean, what they do is they call a couple of pundits and they say, what do you think about this trial? And they say, oh, it looks good. This is the efficacy. And they don't mention the so the press doesn't get it.
That would constitute them having to read. We don't do that.
Right.
That's too much work.
That's dedication. Streaming before she's fully moved in. Everything's in boxes, right, Amy?
Yeah, it's all in boxes. So yesterday I bought a watermelon and I had to spend 45 minutes going through the box to find the box that was labeled knives, green alien, and waffle maker, I think.
Green alien?
Yeah, my Leslie green alien, the Q-tip holder from Jonathan. Can't go anywhere without the green alien Q-tip holder. It's not home if you don't have a green alien Q-tip holder. It's
So
just the way
John
it works.
says Comcast support is disappointing at best. Owning your own network equipment is much better. Yeah, so I told Amy to buy a modem. This is funny. Amy brought a modem and router from New York, and neither worked there, and she had to go out and buy new ones. But then again, you did buy them 15 years ago, right?
No need to focus on the details. No need to focus on how old they were.
Yeah. Okay.
God. Well,
All
the
right.
funny thing is that it all worked 5 minutes until 5 minutes before I left New York when I unplugged it and put it in their boxes and put it in the car. Because I was very smart, right? Because I said, okay, I have to take this last, and I should not put it in a box in case the movers break it or lose it. It should go in the trunk of the car. So it next went to the suitcases. So it was all perfectly fine 5 minutes before I left New York City.
Different company, they have different compatibility. It's fine, but it's all working. What we got you is good.
Yeah,
Do
super fast.
do bacteriophages share a common protein that generate IG that makes them ineffective for phage therapy? I don't have a common protein, but they're very immunogenic. So yes, you make antibodies, and that limits their utility. Yeah, for sure. Who coined the term reverse transcriptase? It was not Baltimore nor Temin, right? Correct. Did you listen to 50 years of reverse transcriptase, the last TWIV? So it was coined by a Nature news writer. So when they wrote the news piece about the papers that Temin in Baltimore had published, that writer, who I don't know who it is, but they announced it at the end of the meeting. That writer coined the phrase. And I didn't know that. That's quite interesting, right? Chevy Chase is a very funny neighborhood. What do you think? What do you think?
Yeah, I do. I haven't seen a lot of his movies though.
What
Like I
kind of
never
car?
saw Caddyshack.
What kind of car did you get?
A Prius.
Which one?
Oh, there's more than one?
I don't know. Don't ask. I don't know what's going on.
I don't know. I type in Prius and that's what vomited out. That's what
So
I put.
Friendship Heights is in D.C. Is the Chevy Chase where Amy lives in Maryland or D.C.? There are two. I have a Maryland address, right?
I have a Maryland address.
But you would like to someday move to D.C., right?
Yeah, when I adult, I'm going to buy a brownstone in DC.
When you adult?
Yes.
You are an
When I
adult
find,
already.
when I have my permanent home. Now I'm just a wandering Jew, as you like to tell me all the time.
No, you coined that term. I would never call you that. That's not nice. But you would like
Okay.
to get a brownstone in D.C., right?
Mm-hmm.
You better get it now before they go up in price. The one you showed me, where was that? In
DuPont Circle DuPont Circle.
three-story brownstone, beautifully refinished for $800,000. You couldn't touch that in Manhattan for less than
No.
$2 million. It's unbelievable. Unbelievable. Do we know how much heterogeneity there is between different people's innate immune system?
Not really.
But there is heterogeneity, right?
For sure. That's
I
what
mean,
Casanova investigates.
there certainly is genetic polymorphism, but whether that's functional or not, we don't know. As you know, SNPs, single nucleotide polymorphisms in innate immune genes are aligned with severe COVID. Besides making
Yeah,
antibodies
so who
against
cares?
interferon.
I mean, the truth of the matter is nobody gives a shit about that. It's the stuff like the TLR SNPs that regulate herpes encephalitis, and he has the UNC93 or 98 in the same pathway that does the exact same thing. That's what he's most,
Yeah.
And the TLR3 SNPs also regulate meningitis or encephalitis that associates with Entro 71.
So Patricia lives in Northern Virginia. You guys,
That's cool.
You should go out and have
We
a
can
drink
stop
sometime.
by.
Yeah.
Yeah,
should
for
have a drink.
sure. For sure.
Lots of congratulations. People are excited about the FDA.
Yeah, it's cool.
What is your prediction for next-gen COVID vaccines and treatments? Any exciting new vaccines in the works? Well, if Amy knew, she couldn't tell you. She's confidential, right?
Yeah.
But I could tell you if I knew, but I don't know. But apparently there are antivirals in the works, which are more exciting than the existing ones that have been told. But I don't know what they are and who's developing them. I don't know about vaccines. What's going to be happening?
Well, there's definitely some rumbling about making an variant-specific one.
So Moderna and Pfizer certainly have an Omicron mRNA vaccine, right?
Mm-hmm.
I don't know which vintage of Omicron they made it against. But yeah, there's some rumbling that in the fall we'll see that. But I don't know. I don't know what's going to happen.
We'll see.
Yes, and we knew her when. We knew Amy when she was a lab rat.
I will always be a lab rat.
And the FDA is better for having Amy, that's for sure. That is for sure.
Hopefully they think that too.
Looks like everybody is happy for you.
It's good. I worked hard. I'm very excited. Couldn't have asked for a better set of colleagues and stuff.
And you are working for Uncle Sam now, right?
Yeah, technically, I guess.
Yeah. I hope. I've never worked for Uncle Sam. I hope it all works out.
I don't see why it's any different than working for Columbia, to be honest.
Laura and Kip, who came to the incubator party.
Yeah, the two farms from San Francisco.
Yes, they're great. It's great to have someone good at the FDA. There are many good people, as Amy says.
Yeah, my colleagues are great.
Mazel Tov Plus. I'm just scrolling through all these lovely congratulations.
Yeah, it's good.
Harry says, is the reason there's no benefit to variant-specific boosters because of original antigenic sin? What do you think about that, Amy? That's not the issue?
No. That says that it makes the disease worse. There's no benefit to the variant-specific boosters because you're still targeting the exact same epitopes. And many of those epitopes don't change, whether or not they're what did Theodore and Paul call them? The class 3 and 4, and then the conserved epitopes of T cells, right?
Correct.
So it has nothing to do with antigenic sin.
A scientist being able to concentrate on science, what a concept. Congratulations. Thank you in advance for the new knowledge you will bring to the world.
Yeah, I'm excited.
I like this one, Amy Unchained. You're unchained from me, right?
Apparently so.
I don't think that's how it worked, Mark. I think she was the boss, really. Right?
You're just figuring it out now.
Yeah.
He's so slow.
Michael says, I wish I could apply to be your lab tech.
You could. The FDA is actually having, they have a call for technicians, and they can just go to the CBER website and look up research associate, I think is what it's called. Hold a second as I flip the mouse around. Hold a second. So if anybody would like to apply to be my technician, they can fill out they can fill out all the information for the ad. Where is it? Hold a second. So it's the CBER an job announcement. It's VIN 1151150 It's research biological scientist.
Let me type it out on the screen for you. So give me the number again.
So it's called the CBER Job Opportunity Announcement. And it's VIN.
Wait a minute. Opportunity. VIN.
115.
115.
115.
Another 115?
Yeah.
Okay.
20.
Mm-hmm.
And it's the research biological scientist.
Okay, we'll put this on the screen. There you go. VIN stands for Vehicle Identification Number. Did you know that?
No, but you learn something new every day.
If you want to apply,
But I do have to say, I filled out that number on multiple forms too.
hmm so if you want to be Amy's technician, there you can go look it up on their website, right?
Yeah.
And this is funny. Mark says, "I will request every biological you research, whether I need it or not, Amy."
Yeah, it's cool.
People like you, Amy.
It's good. I like them too.
I've heard secondhand the CDC and I don't know, NIH.
NFL. I don't know what
Oh,
the
this is
NFL is.
National Football League. Did a study
Ah,
concerning contact tracing and infection. Turns out transmission only happened indoors and no infections from on-field play. I don't know how they would determine that, right? How would you distinguish between an infection indoors versus on-field? It seems to me, and you're never going to see this published because the NFL will keep it quiet. So I'm very suspicious, right?
okay. I don't know.
I think the move has refreshed Amy. Do you feel refreshed?
You tell me, was I refreshed yesterday or was
Ah,
I
she was
stressed
pissed
out
off
beyond
because
belief?
she didn't like driving to all these stores. She likes to either walk or take a bus or subway. She didn't like going 3 times to Best Buy. She didn't like being 8 hours on the phone with Comcast. Yeah, she didn't like it. But now that it's all set up, she'll be good. She's definitely more relaxed than she has been in the past 6 months. Yeah, for sure. Vanity Nutrition says the Prion lecture was my favorite. That's one of my favorites, too. Yep.
You really like transformation.
I love transformation, yeah. Transformation and oncogenesis is probably one of my favorite lectures. And then maybe unusual infectious agents behind that, yeah. A&V, any thoughts on the University of Washington Medical School vaccine? What vaccine is that, Amy?
I think
Do you
it's
know?
alive attenuated. I think it's alive attenuated. And I think it's a good idea. Considering the fact it was what we proposed.
A COVID vaccine has been developed. Here's from the University of Washington newsroom. Safe and effective in large-scale clinical testing. It's a second-generation vaccine, stable without deep freezing.
Oh, I guess I was wrong. Okay,
No.
I was wrong. It's not alive attenuated. That was.
But what is it? I'm still trying to find what it is. They did a phase 3 study in 4,000. Oh, it's made of proteins that form tiny particles studded with fragments of the... okay, so it's a protein vaccine.
Cool.
David Wiesler is...
Yeah, he's a colleague
on
of
it.
Jesse Bloom's. He does a lot of stuff.
Okay.
Very into the evolution of the virus.
This is basically like Novavax, the Novavax that never appeared, right?
Apparently. It's hard to know since it's never appeared.
Vincent, you discussed with Daniel an interesting paper on COVID sniffing dogs. Authors did PCRs on sweat to identify cases. How does fragments of viral RNA get into sweat? What do you think, Amy?
They float through the blood and your sweat, you have sebaceous glands and probably get processed and then they escape out just like, sweat has urea in it and various other things.
I bet you could find a lot of viral genomes in sweat when you're infected, for example, right?
I would assume so.
Yeah, just that people haven't looked.
Mm-hmm.
Look at this. We have 470 people tonight. They came to hear where you're going, Amy.
Cool.
Amy, will you do back assays?
Of course. I'm a little disconcerted that there's no sink in the tissue culture room, but we'll figure it out. Not a big deal.
Is there
Ooh,
any is there
gotta
any
fix that.
is there anyone else at the FDA who does plaque assay?
I think Chrystia does. I think they used to.
Yeah.
Um, let's see. I'm not sure that Gabriel does. And I believe that the mumps, I believe that Steve Rubin did before he left. He was
Mhm.
mumps. Um, Marion, you can't do plaque assays because she's works on hepatitis C. But Hannah Golding she's a retrovirologist, but she now has transitioned to doing SARS-CoV-2. So I would imagine that they do either a pseudotyped plaque assay or have BSL-3 capacity or privileges.
So
So
you
yeah.
figure in a couple of weeks you'll be able to do your first plaque assay, right?
Yeah.
Maybe I need to find a home for the wall of polio.
Not by me.
No, I didn't say that.
No,
Somewhere
we don't
else.
need to find a home for the wall of polio. The
I
wall of
throw it out.
polio is done. Yeah, we're done.
Okay. Yeah, I told you folks, she's the boss, right?
We're done with schlepping the wall of polio around.
Any thoughts on how you would identify a virus that does not cause the disease? That's metagenomics. You just take samples. You could take blood, feces, saliva, um, whatever you want from people and just sequence and see what's in there. You could do the same with animals, and people do that all the time. And yes, for some, we think viruses are beneficial, but it's hard to prove it in people. There's some experiments in mice that suggest it, but don't prove it. But
That's what Ian does.
we have,
Ian used to do metagenomics for pathogen discovery.
yeah. I mean, we think they're beneficial because we have a lot of viruses in us that do not cause disease. And so the idea is that they may be beneficial, but proving it is hard. It's quite hard.
It
Yeah.
could just be nothing.
They could just be nothing. They could be noise, signifying nothing, as Shakespeare said, right?
Yeah, because the receptor isn't present. Like all the plant viruses, they're in us, but the receptors aren't present, right? So they're like noise.
And so the sound and fury signifying nothing. That's from Macbeth. Do you remember Macbeth,
Yes.
Amy?
Out spot. Out damn spot.
Do antiviral mRNAs translate to more than one protein without involvement of a protease? Oh, yeah.
Yeah.
This is Amy's specialty. She could tell you. In fact, the coronaviruses do, right? Frame shifting.
Frame shifting, uh, stop starts, ribosome shunting. Mm-hmm.
Internal ribosome entry sites.
Yeah. Slippery sequence.
Yes. There are quite a few examples, Doug, of that, yeah. So Patricia says, I will miss New York restaurants for you. Northwest D.C. is not great for food. Is that where you are, Northwest?
Yeah, I'm in the Northwest. It's okay. There's this thing called the Red Line, and it takes you to other places in DC where there's better food.
Should we still have the mask mandate in hospitals and doctors' offices? What do you think?
Sure. I mean, you can never tell who's coming in, right? They could be immune compromised going to the doctor's office or the hospital, right?
Yeah. For sure. I agree. Our tax dollars well spent on Amy.
Yes. Give me more money.
So Lisa did her dissertation research and her daughter is moving there. Your talent is needed.
That's great.
The Ernst lecture was most excellent. Be sure to catch it. It's on my YouTube channel. You should check it out. They did a really great job filming it. Yeah.
Yeah.
My grandson has COVID. He's 8.5. The pediatrician gave a prescription for antibiotics for the ongoing
not
cough.
good.
No. Just listen to Daniel Griffin railing about you don't give antibiotics for a virus. And the cough is from COVID, not from bacteria.
Not good.
Now that there's not much to talk about when it comes to COVID, these other YouTube stars are turning their attention to monkeypox. And I must say, they don't know anything about that either.
Oh, that was the other thing we discussed today on the phone.
What does Ian Lipkin think about monkeypox?
Oh, we didn't discuss what he thought about monkeypox. We discussed who was tweeting about monkeypox.
Oh, here's Steph. Steph is here. Thank you,
That's
Steph,
great.
for moderating tonight. I didn't see you before. And thank you, Mark, for your contribution. He wants you to get yourself a bagel.
Okay, well, you can bring me bagels from the thingy, but don't bring any of the cream cheese. It's too scary. You can bring me the pseudo bagels. The place downstairs is just pseudo bagels.
If you want.
They're really big, like rolls. But yeah, you could bring pseudo bagels. Or you can go all the way uptown to Zabar's, get me coffee, and bring bagels and sturgeon.
Someone's just said, Tony, maybe the antibiotic is to prevent secondary bacterial infections. Well,
not a good plan.
No, you don't do that. This is not an issue. You give it when there is an infection. Anyway, don't listen to me. Listen to Daniel Griffin. He's always railing about giving antibiotics for COVID. It's totally inappropriate. I'm just echoing him. I had a third dose vaccine 8 months ago. Want to travel, want to get a fourth dose. Will it ruin the efficacy of a new vaccine if it comes out in the fall? No, not at all. You can get a fourth dose and travel your heart out. Absolutely. And yes, Dr. Griffin had a monkeypox update last week. That's what you should listen to, not the pundits on YouTube. But they're getting listened to by their millions of listeners. I don't know what to do, folks. Amy has to work on a bagel-based OPV. Her daughter is in the hospital with COVID. She's 24. They're refusing to give her remdesivir. Anything I can do? Well,
get monoclonals.
You could. Why are they refusing?
Isn't that the order of things? You're supposed to get Paxlovid, then get monoclonals.
Yeah, but hospitalization is probably too late for Paxlovid. But if you get it early enough in the hospital, before the ICU phase, you can use remdesivir and it's more effective. But they're refusing to give it to her. But I don't know how far she is into the disease. If she's in the inflammatory phase, then she needs steroids, right? It depends where she is in the course of infection. If you want help from Daniel, he will help you. Daniel at microbe.tv. He'll give you.
He'll answer your question.
He'll answer your question. He'll help you. Okay, Jennifer. Daniel at microbe.tv, and he'll get back to you very soon. Why monkeypox now?
Actually, we've seen monkeypox for a while. The thought is that we're now more of an immune naive population. It's always been there.
Yeah, so ever since we stopped vaccinating against smallpox, we've seen more and more cases of monkeypox. Most of them were in Africa, where the reservoirs exist, the rodent reservoirs, and periodically cases were exported to various other countries.
Did you read the science article that I sent you?
I didn't. I just saw it tonight, and I'm going to put it in the show notes for Friday because we're doing an update. But what's the gist of that?
Africa is going to teach us everything about monkeypox and that we should vaccinate, I believe.
Well, they don't vaccinate in Africa, so what are they going to teach us?
So it's the monkey. It's so it tells you it's been steadily increasing for the past 3 decades. Blah blah blah. Demographic shifts. Blah blah blah. Kind of a self quarantine, and they deal with it by let's see. lack medicine to treat. So yeah, they offered the vaccine produced by Bavaria Nordic.
Yeah. Well,
That was generated in 2019.
it's not surprising
And they
that
also
case.
give us experimental drug that was launched by Oxford in 2021.
So in the U.S., we have a Canvas 2000 smallpox vaccine stockpiled. We have 2 antivirals stockpiled if it's an issue. Not surprising
So we
that
should get that out.
it's not surprising that the cases are increasing because more and more people are being born and not being vaccinated with smallpox vaccines. So we have a bigger and bigger susceptible population.
That's what I just said.
And why now? There's never a good
Because
answer
it's
for
even
why
bigger.
now.
It's even bigger. Because it was airborne and it's even bigger. Because of all the masking and quarantining. So it's even bigger.
That
The
could be, yeah.
naive immune population is even bigger.
It could be. Thank you, Morris, for your contribution. Appreciate it. Is it known why smallpox is exclusive to humans while other orthopoxviruses have an assortment of hosts? No. I'm not sure we understand that. But you're right. There are a lot of different kind of orthopoxviruses with different host ranges, but we don't really understand that.
There's a lot of viruses that are only human specific, have specific host and a lot of host ranges. Measles. How many people?
Well,
When was the last time you saw a cat walk around with measles?
so polio is specific to humans, but many of the enteros infect other animals, right, Amy?
Yes, 29, 99, others. But when was the last time you saw some cat walking around with measles?
Amy, on your first day at the FDA, why don't you wear a T-shirt that says, right, Amy? Just kidding. It's a joke.
I know. I laughed.
Amy, do you
I
have a
raised the
backyard, a
eyebrows.
big house?
No, I don't have a backyard or a big house. My apartment comes with a rooftop pool.
Amy is viewing this apartment as a temporary thing, right?
All apartments are temporary in my book. Then you get the big house. Then you get the house, or you buy something, whatever.
Mike
House,
Ryan of
apartment,
WHO said today that climate change has increased the frequency of endemic diseases such as Lassa and Ebola. I don't see how you could make that statement. Do you think you could make that statement, Amy? Causation?
Yeah.
I don't think you can make it. Maybe it's something else.
He didn't say caused. He said increase the frequency. He didn't say caused.
Climate change has increased the frequency. That's a causation.
Okay.
I
Whatever.
just think that I don't see the data for that.
I'm not going to argue with you.
Usually you do.
I'm too
Who
tired.
do you view as the top virologists in the UK? I'm not going to name names. Do you know any top virologists in the UK? I can't name names because then the other people will get mad at me, so I'm not going to name any of them. Is that a good strategy, Amy?
Sure. Move on to the next question.
Okay. In your lecture, why did you discuss transmission by droplets and not aerosols? I did discuss aerosols. You didn't hear it, but it's there. I guarantee it. I've been giving the lecture for 15 years. It's there. And yes, we have an expert at Harvard who apparently, I don't have an assistant, but who helps me with programming, but we have an expert at Harvard who I will get on to talk about droplet transmission. Is there a reason This Week in Neuroscience isn't on Spotify? Okay, so I went to Spotify, and there's a thing that you can do to add a podcast. And I went to add it, and it said it's already there. Sorry, bub, it's already there. And I don't have time to deal with it. So if it's not there, I'm sorry, but at some point, maybe I'll get my non-existent assistant to take care of it.
You could have Hannah look into it next week when she's more established.
That's a great idea. Let me write that down. Oh, folks, by the way, I ordered my YouTube 100,000 Platinum Award. It's free, but you have to order it. I found the link. Someone had nicely sent it. Yeah, it's going to, and then I'll do the unboxing here on the live stream. Hannah, what am I going to do with her? Do what?
Twin on Spotify.
Thank you. May I suggest climate change has very little effect on movement, plant, and animal life compared to intentional introduction of plants and animals and large-scale shipping. Well, yeah, so far it's started, and according to this paper, when it really gets going, it's going to be massive, because animals don't like to stay where it's uncomfortable for them, right?
Right.
And Dixon seemed to buy that.
What do you mean seem to buy? He was in agreement. They move where the food is.
That's what I meant, yeah.
He said, move to the food.
They don't have the background education or training to critically evaluate the trial data, so they have to rely on people who do, like you two. But they don't ask us, Harry. That's the problem. We have our own shows, and we reach a lot of people like you folks, but the press doesn't talk to us for the most part because we're not pundits. It's fine. I don't have a problem with that. I have all our shows, and we have this live stream. Thank you for getting, for Dr. Griffin, for the info, helping me through my first infection. Good. I'm glad it worked out, Penelope. There was this is for Vern. There was a recent article about the growing movement of life into elevations. Okay. Let's see. Do chat questions sometimes give you new ideas or thoughts? Yeah. That's what I like. That's why I like doing them, right? I find them challenging, and definitely we get new ideas. Don't you, Amy?
For sure.
Yeah, I love the chat. If we didn't love it, we wouldn't do it, right?
For sure.
Mark says you should move to Georgetown. It's pretty expensive, isn't it?
I don't know. I don't know exactly where it is.
It's a good neighborhood, but I think DuPont Circle is good too.
I didn't move there either. I moved where there was an apartment at the time that I needed an apartment. That is what I did.
You are radiant tonight. Thank you, guys. If HPV vax doesn't prevent infection, how does it prevent cervical cancer?
It prevents... Well, go ahead.
It prevents... It allows infection, but then the antibody, the memory response limits the infection. The CTL response limits the infection, so you don't get the extensive replication. The cervical cancer is caused by aberrant integration of the viral DNA into the host chromosome, and you don't get the extent of replication that you would need to do that. It's a rare event, so you need a lot of replication to see it, and the vaccines prevent that. So they limit the amount of virus replication. Okay. Carolina says she can relate to the wandering Jew. Okay. Amy, we have no congressional representation in D.C. You may really, really, really miss it. Prices are already way up. And here's your buddy.
Really? Like it was cheap in New York City? Oh, it's Animal Party. It's Jen. Wow.
Yeah, Jen probably
Cool.
missed your announcement, but she could find it.
Cool. Wonder how her bees are. You know, she's got bees.
I didn't know that.
Not just her little quail. She got bees too.
Vincent, Prince. Oh, so Nama, I thought I had shipped them all out. Please send me your address. I will ship out your book, Vincent@microbe.tv. I actually have one copy left here, boxed up, ready to go. Get me your address tonight. I will ship it out tomorrow. What do you think about that? Sorry.
I'm still flabbergasted at the efficiency. I'm speechless.
Thoughts on Evusheld?
Talk to Daniel. He seems to be he seems to like it.
Remember, Evusheld is a preventative. You take it before you're infected, and it lasts for up to 6 months. And then you need another bolus. And he's very bullish on it. That's the word, right?
Mm-hmm.
How many hoops did Amy have to go through to get the new gig? At first, you had a telephone interview, right?
No, I had a Zoom interview with the whole FDA.
You gave a talk, right?
Yeah.
And then you went in person, right?
Yes.
So that wasn't too bad, right? Not too many hoops, no?
It's good.
Is SARS-CoV-2 vaccine going to be seasonal like influenza? Well, that's the question, isn't it?
Most
Well,
likely.
I suspect it will, because this is going to be a seasonal infection. We're going to have it every winter, forever. So if the current vaccines don't last long enough, yeah, you're going to be getting new ones. What will you be doing in your new job, Amy?
Research.
On
Review.
what? Enteroviruses, right?
Yeah.
So Amy's been working on enteroviruses at Columbia, and many different questions, but the most recent one, the cross-reactivity of the antibody response, and that's something she wants to get up and going right away. And that's completely dovetails with the FDA interest. It's perfect. It's really perfect.
Yeah. It's good.
And so then as she gets more people, she can do some other things. She's got a lot of other projects in mind. So you'll
Well,
hear
we
as she
need to
publishes
develop
them.
the animal model. We need to develop the immune competent animal model.
She wants to develop an immune competent animal model for acute flaccid myelitis caused by enterovirus D68.
Well, we need for other ones like C99 too. The question is whether or not they all cause it the same fashion because it's just a clinical diagnostic, right?
Okay.
You want to know mechanistically if it's all caused by the same way, right?
Right.
That compares 68 to 71 to polio to C99 to D94 and blah, blah, blah.
Mm-hmm.
Could be really interesting because it could teach you a lot about neuroscience.
Yeah, it's going to be very exciting. Basic
Yeah.
virology. How much sway
Basic cell biology too.
how much sway do pharmaceutical companies have on what studies get published?
Not that I'm aware of.
No more than anyone else, unless they're reviewers, right? Sitting on the reviewing boards for journals, they don't have any more sway than anyone else does. No. Do flu vaccine makers have to retest their safety every year when they adjust the strains? Yes, they do a very small safety assessment of 20 or so volunteers, and they do safety, which is not a big amount, but they do it, and then they measure antibody responses as well. Amy, I recall your pick of the premonition. I read The Fifth Risk, also from Michael Lewis. Thank you for such a voice of reason. Do you think we will appreciate science and perhaps government?
In this lifetime?
I guess the answer is no.
Call me back. I'm not bet. I wouldn't. I mean, I'm not a betting person, but I think it's kind of like 88 to 1. Although the 88 to 1 horse did win the Kentucky Derby, so there's always that potential chance. But I don't think lightning strikes twice in the same year.
Omicron 4 and 5 taking over Omicron 2, etc., more immune evasion. If Omicron is no less severe, then are we headed back to square one with infections and ever more evasion? No, because as long as the disease doesn't increase, the immune evasion isn't an issue, right? So far it's held up.
Who cares?
But if severe disease increases, then they will give a variant-specific vaccine. Animal Party would love to work for you. I'm so jealous of anyone who gets that job.
She's going back to school to become a nurse practitioner.
I heard.
Mm-hmm.
I heard.
It's very exciting.
Put that in the show notes, please. Lab tech job listing. I don't have show notes for the chat for this live streamy thingy, but I will add it to the chat, or I'll put a comment in tomorrow. Yeah, that's fine. Okay. Besides influenza vaccine, is there any other which could induce a P24 antibody and immunoblot? You're talking about HIV P24. I don't know about influenza-inducing antibodies to P24. That's
Well,
HIV.
there's a lot of things that run at 24 kilodaltons. That's why you use an antibody.
Does the YouTube know you do these? Does the FDA know you do these? Are they going to make you stop? I know sometimes they're weird about that stuff.
We'll discuss. I'm sure that we can. I'm sure that they know. Sure, we'll discuss.
And Amy hasn't started working there officially, so this is okay. And if they say, Amy, you can't do it, then it's just me, right?
Apparently so.
And then I'll lose half the people. And then the next day I'm going to get a call from Amy. You said this wrong. You said that wrong. You said this wrong. You're messing up my show. Stop it. I'm just playing.
Pretty much. No, pretty much that's how it goes.
John,
don't mess it up after I built it.
It's been a stellar few weeks here at a Microbe TV. It feels better to contribute than to invest in for-profit ventures. We're a nonprofit venture. You don't get a return on your investment, but you do educate the world. So it's a good thing. Why is poliovirus so stable, Amy?
Stable how?
Thermostable, for example.
It's by the chemical bonds in between the amino acids that form the beta sheets.
Yes, this capsid structure is beautiful.
Well, it's
beautiful.
it's not. There are a lot of things that are icosahedral that aren't as stable.
Yeah, of course.
And so it's the amino acid residues and where they and how they interact that makes it so stable.
Yeah.
So
they're using Novavax in India. It's called Covaxin, I think. FDA is reviewing it this week. Okay. That should be interesting.
Yeah, for sure.
I have an impression that an inactive vaccine is bad at eliciting T-cell responses, but I also recall that antigen-presenting cells can do MHC class switch. Then it should be able to activate T-cells. But APCs can activate T-cells. That's how they query. APCs present antigens to T-cells, and that's how they query whether they're foreign or not. So why inactivated vaccine is poor at that, I don't think we understand. Are you going to stay the whole time tonight, Amy?
I'll stay till 10. Then I have to go eat dinner.
Me too. Oh,
you didn't eat dinner?
No, I didn't. I got home late. The train was late tonight.
Wow. What are you going to have for dinner then?
I don't know. Maybe scotch.
You can't just have scotch if I can't just have gin.
Who said you couldn't just have gin? You can have gin if you want. No, oops. All right, Andrea, correction. Covaxin is the Bharat Biotech inactivated vaccine. Right, I thought so. The NVX is called Covovax in India. Okay. Are they going to make COVID vaccines for dogs and cats? What a good question. What do you think, Amy?
No.
They're not? They're not going to make vaccines for pets?
No, because they don't really display disease.
Okay. I got an MMR booster in June. 1 Pfizer dose. Haven't had COVID yet. Probably just luck, but I keep wondering. Mother and Sibs haven't had it either. Yeah, it's luck so far. You maybe, depends where you live, whether there's, maybe you're in a place where there's not much virus, but you might not want to take your chances unless you're going to not travel, right?
Mm-hmm.
Maybe you could move the wall to your basement studio.
No.
Gonna move it without telling Amy, okay? There's no way she's gonna find out. No way in hell Amy would find out.
Yes, because what I'm going to hear is Doris is unhappy with what I've just done.
I'm not going to move it here. No, I wouldn't move it here. There's another place I could move it.
Really? You have a second basement studio that we're all unaware of?
Oh, this is it.
Because it's not going to the incubator studio. And so
You'll
it's not
never
going
know
to your basement
because
studio.
You never go to the incubator anymore. You would never know.
Believe me, I will know.
Oh boy.
especially since I have to come for fundraisers. I will know. Do not think I am not omnipotent. Do not question my powers. Some
Novavax vaccine
people,
is being used across continental Europe and parts of Asia, big in France. Cool. I didn't know that. I didn't know that. Any more updates on monkeypox in the genome? Any changes that might make it more contagious? Got into a huge fight with Reddit and got banned because I was sick of the fearmongers. Yeah, immediately they're going to blame it on mutation. I got news for you. This is a DNA virus. It mutates far less than RNA viruses. And these monkeypox isolates, you can go to nextstrain.org/monkeypox and see all the sequences. They're all almost identical. And they're not very different from the 2018. What was it? An African isolate, you said, Amy?
Israel.
Israel. And so there's no reason to say that it is mutated and that's causing this pattern. It's absurd. It's the same dumb conclusions that everyone makes whenever a virus seems to be doing something different. And you got into a good fight, Eddie. That was a good fight. I'm sorry you got banned. I should do one of my weekly Reddit thingies, Amy. Maybe next week
I
will
don't know
do
why
it.
you stopped.
I'm going to put it on my calendar for next Wednesday, okay? Wednesday, June 8th, Reddit AMA. How's that? Okay?
Sounds good.
From 11 a.m. to 1 p.m. If anyone wants to check into Reddit, thanks for reminding me, Eddie. That's great. How many sequences would you have to run to have sequenced all the virus in your fingertip? You do one run on a Illumina thingy, right, Amy?
Yeah.
It would take half an hour, really?
Mm-hmm. No, it wouldn't take half hour. It would take like half a day.
Oh, by the way, put the wall of polio next to your guitar. No, but what if I put just a short wall of polio over the mantle, Amy? You know, see the mantle?
Over across the Michelle Banks picture.
Yeah. I could do like
across the Michelle Banks picture.
A 4-layer plate.
No,
I could I could raise it up and have a 4-stack of plates. Just kind of a reference to the original wall. I think this is a great idea.
No.
I think it's a great idea.
If you get to no, I don't think Darius is going to be happy.
Can you explain the process?
You're not coming to live with me.
I know.
So you have no choice. You have to do what Darius says, and I don't believe she's going to say that's okay.
Can you explain the process of putting ACE2 receptors in mice? So there are 2 ways that it's done. And the first is to make a transgenic mouse where you take an ova out of a female mouse and you inject the ACE2 DNA into it, and then you put it back into a pseudo-pregnant mouse, and then some of the offspring will have the transgene in their germline, and you breed them until you get all the mice to have the transgene. Those are transgenic mice. The other way is you could put the gene in a vector and infect the mice. So one way is to put it in an adeno-associated virus vector where you get long-term protein production. You could spray it into the lung and then infect. And that was done on one of the recent papers that we did as well. And I have to tell you that the first research group to make a transgenic mouse model for a viral disease using a virus receptor was a group at Columbia University. They took the human poliovirus receptor gene and made transgenic mice and showed that they could be infected with poliovirus. That was the first time that was ever done. Is that all good, Amy?
Yeah.
Amy has a moth in her house, I guess. But now you messed up the camera, Amy.
I'll put it back. It's fine.
Was that a
Had
moth?
to kill a... No, it was like some kind of fruit fly. But I don't have any food in my house, so I don't know where it came from.
It's from your neighbors.
My neighbor has a 3-year-old that sits on the balcony a lot, so maybe, but I haven't opened the balcony door, so I don't know where it came from.
Is there any
But
viral communication similar to bacterial quorum sensing that could become a target for disrupting population functioning? So interesting that you should
yeah,
ask, Ray. And, we have done a couple of papers on TWIM, which indicate that virus-infected bacteria secrete molecules that can be sensed by other bacteria. So there's something going on like that, at least in
but even
phages.
Jim Gurns stuff. So like Jim Gurns has like where you infect with rhinovirus, and then it really causes the release of some metabolite that uh, recruits myxoma, myxoma bacteria to make
Hmm.
like a biofilm. It's pretty cool. Can't wait to see how he teases out all the genetics and the mechanism.
I like this. Jon Snow's rules in current
We should
monkeypox.
talk to Ian.
One, all early data are wrong. Two, never trust weekend holiday data. Rule three, don't assume anything. Rule four, double, triple check your denominators. I think, Hans, that's all right, and this is not what most people do.
Talk to Ian. He's the John Snow professor of epidemiology.
I
No,
have, yeah. Did you talk to him about monkeypox today?
not scientifically. We talked about who was tweeting and who was going to screw it up.
Tweeting is a waste of time.
And we all decided that the same person who has been tweeting for SARS-CoV-2 is going to screw up monkeypox.
I know who that is.
Really?
Yeah, Sean has a bad cough, but the home test came out negative. Well, you might not have COVID. Other things cause cough, right? So if you want, take a second test the next day, but you may not have it.
Yep.
All right. Could the new monkeypox be connected to Wuhan? Sure. Every new virus comes out of Wuhan. No, not at all. If one is exposed to SARS-CoV-2, does one create IgA antibodies on top of the IgG from the vaccine? Do we create IgA faster upon being reinfected? All right, so I'm not quite sure I get this. You're saying you're vaccinated and now you're exposed. Do you make IgA? That's a good
Yes.
question. You think you would make a memory response, Amy?
There's there's IgA. Yeah.
Yeah, I think it depends how. But what if the vaccine is limiting the amount of virus reproduction? Is that going to still give you an IgA response?
I think so. Plus, you're actually getting infected in the upper respiratory tract where there's no IgG.
The IgA memory cells, though, are not long lived in the tract. That's part of the issue here that I'm struggling with. But I'm not a
Well,
secretary.
but the IgA is what is protective in that, and is typically what the response is in the upper respiratory tract. It's not an IgG based response. So if you're
So
arguing that the memories are short lived, well, then you should get an IgA response.
yeah, the IgA memory is short lived as well. So you're saying there's no IgG in the upper tract?
Right.
So what limits infection after vaccination?
It has to be part the innate immune response and part the IgA.
Mm-hmm. Okay, and then
But when we
eventually,
investigate that, we when you actually do investigate that, then we'll have the answer, right? But IgG, you're just monitoring serum levels, and
correct, yeah.
so I don't know what I don't know what the claim is, and it's protective from the development of severe disease because it's found in the lower tract. So it's fine.
Why might the body produce hives a couple days after a fever from a presumed fever? My little guy just broke out in them after a battle with a bug. Thank you. Inflammation?
I don't know enough about it. Talk to Daniel.
Yeah, I mean, I think if you have an infection, you're making an immune response, inflammation, and that may contribute to hives. But yeah, Daniel would know. Hmm. This may be a Daniel question, but should I take any antiviral when I reacted to generic Tamiflu with hemorrhaging for a month? No doc can say why it happened. You should
Talk
ask
to
Daniel.
Daniel.
Daniel at microbe.tv, and he will answer it. You know, he has, we're doing a TWIV on Thursday. We always do them on Thursdays, or mostly. So send it in before Thursday, and I'll make sure it gets up there. And
We're going to have more than 4 emails for him to do then.
It's okay.
You only like to have 4.
500 people, 250 likes. Hit the like button, folks, so we can combat the pundits who get more views here and they don't know what they're talking about. I brought the 85-year-old mom to the geriatric center at Penn. She's triple vaxxed. They said, why not 4? They mentioned out of 2,000 infected patients, 4 times vaxxed, only 1 got hospitalized. Well, that's kind of like, what is this study from? I don't even know what they're talking about. But if she's 85, she's certainly eligible for a 4th dose. And here's what a 4th dose is going to do. For 6 months, it's going to prevent infection. And then after that, it's going to be the same as any other dose. But you get 6 months of protection. And if you're someone who's at risk, like you are if you're over 85, that's not a bad thing. 4-year-old's infected with Omicron. How long should he wait until getting his first vaccine dose from the time of infection? 2 months, right, Amy?
Minimum.
Minimum 2 months. There was a case of vaccine-derived polio 2 in Ukraine earlier this year. Has there been research on circulating VDPV field variants in countries after switch to solely IPV or VDPV eventually eliminated? Good question, right?
Yeah.
The problem is...
And yes, there is research on circulating VDPV.
In countries that are just using IPV, that's the question.
They survey all the countries.
So Israel, for example, switched to IPV a number of years ago, and they had circulating VDPVs as a consequence because IPV doesn't immunize the gut, and so you're never going to get rid of
not without exposure to infectious virus.
Yeah.
If you have exposure to infectious virus, then you can get gut immunity from IPV. Marion Koopman.
Do you think a new variant of concern will arise or have the conditions needed for major variant production change since Omicron and nearly ubiquitous population exposure? I don't know if a new variant was going to arise. I have no idea. I mean, Omicron is continuing to diversify, right? So why not? But it's really difficult to predict, and anyone who does is simply guessing.
Predict schmecht.
A&V, I listened to TWIB 468, Zika by the Slice. Do you remember? I do. Amy was on it.
Of course, it's my work. We talk about the Dubin paper, where we made organotypic brain slice cultures from various aged mice, like day 11, day 15, and day 17, infected them with various isolates of Zika. Saw one isolate from Nigeria didn't replicate at all. Then we followed it up with looking at the Vimentin cytoskeleton and also looking at whether one of the consequences of that study was us electroporating our RNA from different rhinovirus isolates into the brain and asking if it could replicate. And we never saw infectious virus. Yeah. I remember all of that.
It's a good title.
Zika by the Slice. Yeah, and I still have all the Zika isolates. And Chumikov did some Zika work with Raul, so maybe we'll do a little Zika at some point. There's
Lisa,
still a lot of questions.
Thank you for your contribution, Lisa. Appreciate it. Garrett's in New York City. How do I find my way to the incubator? Vincent at microbe.tv. I'll tell you how to get there. Would I be able to pick up a copy of Principles of Virology? I don't have any at the incubator lately. I'm sorry, Garrett. Can vaccinated moms pass along long-term protection against COVID through breastfeeding, or is it just temporary antibodies?
Maternal antibodies last a year.
Yeah, they're temporary, not long term. I'm still waiting for the red parasitology book to be shipped. Yeah. Did you win on TWIP? Yeah, I'm a little behind on the TWIPs. I'll get to it. No worries. No worries.
He might want it before it goes out of print.
You think?
I don't know. That's just, a guess on my part.
Any idea why Evusheld is still effective when other monoclonals were evaded pretty quickly by variants?
I think it targets a very conserved epitope, right?
Yeah. It's a different epitope from the other monoclonals, yeah. Animal Party says, my bees are doing so well. I'll send you pictures. I thought she had quails, Amy.
She has both. She gave quails to her mom and she has her own bees. You got to keep up with the
I
lady.
don't know
She's a
anything
hip
about the
happening
bees.
mom.
Sorry, I don't
She's
know
all
anything.
over the place.
Okay.
She and I know about the bees. You don't need to get involved with the bees. Send
Michael
pictures
says,
of the bees.
Here in Mexico, the government wants to vaccinate children against COVID with the Cuban jab, Abdala. But I heard there isn't even a third phase for adults. What are your thoughts? Do you know anything about Abdala, Amy?
No, I know my friend's last name is Abdallah, but no.
Yeah, so that's the Cuban vaccines. The trial results have not been published. But I'm trying to see here if they did a phase 3. Clinical trials. Okay, phase 1, 2, and 2021 phase 1, 2. You're right. They didn't do a phase 3 trial. So what are my thoughts? Not a good idea because you need to know the efficacy against, COVID, against disease, and you need a phase 3 to get the numbers for that. And if you're not doing a phase 3, then they're basing it on antibody induction, which in my opinion is flawed. So I wouldn't do it, but nobody asked me except you. That's what I think. Will there be an mRNA vaccine for flu? Amy, do you think there'll be an mRNA vaccine for flu?
Well, they're trying to create one.
Yeah, they're testing one.
See.
It got a good chance of working, right? We'll see. Yeah.
I think it was less than. I think that there were some issues, but I didn't go
already?
to the talk. Yeah, somebody presented data at a talk at Columbia, but I didn't go to the talk, so I don't really know. But I think that there was some surprising things because they made it elicited a broader amount of antibodies to epitopes, but I think that there were also some concerns. I didn't go to the talk.
As Les says, Moderna and Pfizer are working on it.
Mm-hmm.
I believe you mentioned in Virology Live a warming climate was bringing mosquitoes further north. Yeah, so that's one thing, but also the animals are moving. So it's multiple things happening. You've often said that vaccinated people who get infected might get the sniffles if they notice it all, but lots of my friends have gotten it lately and all have been sicker than that. New variant, not necessarily. Remember, the efficacy is not 100%. So if a lot of people are being infected and it's only, say, 85% effective, then a lot of people are going to get more severe disease. It could also be related to the Omicron diversification. We really don't know. But you have to be careful about anecdotal data, Gene. Just because your friends are doing this or that doesn't mean that that's the trend in the whole country or the whole world. When scientists say they find evidence of ancient viruses in our DNA, what does that mean?
They're retroviruses. They've integrated.
Retroviruses integrated many years ago for Homo sapiens probably, and fragments remain. Does it affect our immune response? Well, actually, some of those genes have been reused for our immune response, and so they are beneficial. And some of those genes are used to make placentas. And so that's how evolution can work. It can reuse genes that it acquires in an organism. Yeah. Ian says the first notes in the horn quartet at the Ernst lecture were CDCGG, which is CD 155 in C major. Yeah, he told me the first two notes were CD. The CGG is 155?
Yeah.
What does that mean? How is that 155?
Don't you know anything about the scale?
Can you just tell me instead of insulting me?
I'm not insulting you. Have Ian explain it to you. I don't want to take his thunder away.
Ian who?
But C
Oh,
is
Fraser.
but usually C is, since you have middle C and that's your orientation on the piano, it's usually considered 1. And then 1, D, E, F, G. It's the 5th letter, so that's the way the scale goes.
Got it. That's very clever of him. Nice. Thank you, Ian. My NP asked if I'd ever had hepatitis vax. If I had one 50 years ago, would a blood test now confirm this? Don't get a blood test. Just get new shots. I presume you're talking about Hep B? But I don't think the vaccine is 50 years old. So you probably didn't get it. And the Hep A vaccine is not 50 years old. So just get a new vaccine, yeah. What do you think of that, Amy?
Yeah, it's not 50 years old.
Could a widespread use of a future antiviral knock out this pandemic for good? Are there antiviral people like anti-vaxxers? Of course.
Not that I'm aware of that. There are not that many.
There's always someone. Well, there are lots of anti-molnupiravir people, right?
Yeah, well, it's a crappy drug. Although I do have to say my cousin had COVID and so did her daughter. And her daughter had no symptoms, whereas mom was like totally knocked out. And they went to the doctor and the doctor in Florida told her there was like very little data about Paxlovid. So even though a different doctor had given it to her 16-year-old son, my cousin was unable to get it. So it was totally ridiculous. So I guess if you're talking about antiviral people, it's like doctors who are ignorant. Not so good.
So principal manager, knock out for good. No, you're not going to knock out this. The virus is always around.
Even if you got a resistant variant, it's not going anywhere. It's an acute infection. That's what Kempfner told you. Pufner, whatever the guy at Emory's name
Richard
is.
Plempner.
Yeah.
Yeah.
See?
So Vern says, I suspect Amy will have to coordinate with public affairs. We'll have to be careful not to mention confidential information and be clear that opinions expressed are Amy's, not the FDA. Yes. And plus she's doing it at home, which is the only way it could be done. But she'll deal with it because Amy is actually quite conscientious.
Yes, I'm very responsible. That's why I lose the car all the time in the parking lot.
Please don't drink on an empty stomach. No, I won't. I was just kidding. And Steph wants to know why you don't have any plants. Amy doesn't do dirt. Amy doesn't do dirt.
No dirt. No dirt. No dirt, no dirt, no dirt.
Send the wall to MoMA. Yeah, I think it's a museum piece because it is my masterpiece. Some museum should want it.
Your masterpiece? It's not a masterpiece of art.
Someone
what are you,
came
Jackson Pollock of art?
A student from Korea.
A
student from Korea came in once and said, "This is your masterpiece." And I said, "You know what? You're right. It is my masterpiece."
So what is all of this?
Can I make the wall of polio a non-fungible token? I bet I could, PM. I bet I could, but it's too much work. I don't want to do it.
Let's not get crazy here. You're not Bitcoin here. And Bitcoin has lost like 70% of its value at one point this year. Like 2 weeks ago, it lost like $3 trillion. And then the other one is like totally bankrupt.
Here's another neighbor. Dave is in Chevy Chase. So
well, he can stop by and we can have drinks on the rooftop pool.
Well, you should go meet somewhere and have a glass of wine.
Okay, so have them email me and have wine. I'm happy to do that. I'd like to
So,
go out of my apartment. I
so
don't know
first the
any place
wall,
to go.
first the wall would have to be digitized, which is not a bad idea in virtual reality. Yeah, you could digitize it. Yeah. I changed my vote, moved the polio wall to the incubator. But Amy is nixing that. I don't know why. I think it's a great idea. Why can't I move it to the incubator?
There is no wall space. That's the end of the discussion. We are not discussing it any further.
I'd want to hear votes from you listeners. You got
No, this is
543.
not a democracy.
A
You are under
republic.
a false impression.
A
It is not
republic.
a democracy, and it is not a republic. It's an authoritarianism. It is defined by me.
So monkeypox is not really a
You are
thing
crazy.
for anyone to be worried about. I haven't paid much attention to it. I think the
True.
outcome of Daniel's and our last TWIV update is that it's not really a concern for most people. It seems to be spreading by close sexual contact in certain populations, which doesn't mean it's not going to spread elsewhere, but I don't think it's a problem. If it did spread more extensively, we do have vaccines and antivirals, so it's not an issue. Hello, Jeff. Nice to see you tonight. Are we on the big screen? The big screen? That would be cool.
Yeah, 72 inches of you and me. It's quite scary.
Jeff was recently at the incubator for our opening. It was great to see you there.
Mm-hmm.
I had COVID with low CT, and 11 months later got J&J 5 months ago. Do you think I need a booster? I think infection in 2 doses is the ideal, right, Amy?
Yes.
Yeah, you should get another vaccine. It could be J&J. You could get an mRNA vaccine. Yeah, I would do that. Okay, now he's changing his vote back to polio in the basement. No, it's not coming here. It's going to go to the incubator.
It's not going to the incubator. Your guitar could gently weep all its want by Eric Clapton. It's not going to the incubator.
All right, so Amy, on the phylogenetic tree of the monkeypox, they have this number, divergence 0.0013. What does that mean? That's distance in bases, right?
Yeah, genetic mean distance, yeah.
But do you know how to interpret that? Is it so many bases per 1,000 or per 100, or what is it?
Per 1,000, I think. I'm not sure. Edmund would know, but I'm not sure.
Could ask Nels. I would ask
Yeah,
Nels.
you can. Sure.
Are the lab mice used for virus research chimeras? Well, it depends what mice. Your standard lab mice are inbred, but you can have chimeric mice.
You have a chimeric. PVR is
Yeah,
on a chimeric
like
background.
polio receptors.
No, it's a C57 times
SJL.
Swiss web. Yeah, exactly.
Our favorite cardiologist is at it again. He's doing more to undermine confidence in vaccines than any anti-vaxxer. Yes, rhetorical question. Understood. The cardiologists in general are always doing damage. What's the difference between transgenic mice and chimeras? It's the same. We use the terms interchangeably, right, Amy?
They're not the same.
We use them interchangeably, right?
I don't.
All right, what's a chimera? A recombinant, basically.
So a chimera is like 2 different genotypes, like the agate mice or chimeric mice.
Okay.
And
So
a transgenic mice has a transgene inserted into it, so it's not the same thing because so like the SCARB2 mice are transgenic, but they're on a pure C57 block 6 background.
Correct.
You just never you just never crossed them over to an inbred background. You left it on a agate. Because Cowder used to get all these mice that were calicos.
Yeah. How would you quantify vaccines' impact on the pandemic? Oh, a total changing of the course of the pandemic, I think. I think we would have had far more deaths. Look at 1918, where we didn't have a vaccine, far more deaths. So I think it's been amazing. quantification. I can't do that, but most of the world still isn't vaccinated, right? But that
Mm-hmm.
is, it's been an amazing impact, in my opinion. So there would be like maybe 100 different types of virus on your fingertip. Would they all be known to scientists? Would there be a bunch of new sequences? There'd probably be new sequences, right, Amy?
For sure.
Yeah.
We don't know half of them. Actually, we probably know about 5%, if that.
Okay. So Jeff and Andrea are
Well,
going to have a little conversation. That's good.
who's Jeff and Andrea?
Jeff, our buddy Jeff Millard, is talking with Andrea about why the cardiologist has undermined vaccination. So I don't think we need to discuss that here.
Yeah.
Okay. I'm only
Oh, it's
banned.
9:35 already. So it's 9:35.
I know.
What do you do now? Oh,
We're
okay.
gonna finish up the questions. I'm only banned off of one of the subreddits. No loss. Only got angry because I'm going on vacation. The last thing I need is this happen. Okay, sorry about that.
I don't even quite understand what the complaint is or what he did.
He said that you can't explain what monkeypox is doing by mutation. There's no proof of that, and they didn't like that. They don't like the
Oh,
truth. Do you think the cases are rising again because the mask mandates were removed?
no.
People are moving about the country, going on vacation, coming back from school. That's what's driving it.
The truth is if we did this for flu, we'd see the same pattern.
Yeah.
If you did this for
Yeah.
rhino, you'd see an even worse pattern.
Of course, is our cases rising, Amy?
I don't know. We can go to the New York Times website now that I fixed my subscription.
Well,
I can
Canny
tell you
says,
right now.
according to the Times, cases are falling.
Well, I can tell you right now. So, oh wow, there's a gunman shooting around with a rifle in a Tulsa medical building. Not so good.
That's not what you're supposed to be looking at. You're supposed to be looking at
New
cases.
cases are down 1%. I don't consider that a big deal. That's like
Okay.
basically they're static.
I want to thank
Hospitalizations are up 10%.
just another human. Thank you for your contribution. Appreciate it.
Very nice.
Is all mucosal immunity around the body more or less equal? For example, would the HPV vaccine be inducing high GA in the nasopharynx? So that's an interesting question. So it's the fact is, the observation is when you immunize one mucosal surface, you do see responses elsewhere, but I'm not sure that they are as strong as you see in the immunized place. Would you say that's correct, Amy?
Sure. Yeah, I don't really know.
Yeah.
I'd have to
So,
brush up. I've forgotten.
and that's been studied in animals, so you can immunize mice, say, rectally, and you get a nasopharyngeal response as well. Okay. Lots of things that I don't want to touch. They want to know about people.
People.
Yeah, various people who proclaim things. And I'm not gonna comment on people who
Proclaim
are,
things. What are they?
like the
Omnipotent?
cardiologist effect. There are lots of
The proclaim
cardiologists.
is not a good proclaim is not such a good word. It sounds too religious.
Here's the
I
Harry says
proclaim.
it all. Cases are falling. They will rise again. They will fall again. You're absolutely right, Harry. This is the trend forever. This is the way it's going to
It's a
be.
trend for everything.
Yes, it is.
Cases of EVD68 rise, they fall, they rise, they fall, they rise, they fall. Cases of rhino rise, they fall, they rise, they fall. I mean, come on.
I
That's
saw
what
a
it
JAMA
how it
paper
works.
which seemed to indicate that vaccinated people were mostly not contagious after 4 days, and naive people was more like 10. Yes, in fact, Daniel mentioned that on his update last week, and he said, "What do you think, Vincent?" I said, "I'm surprised it's not more striking than that." And so I just wonder if the data are It's a small population. I'm just not sure about it.
Well, what test did they use?
Plaque assay. It's good. My primary care physician says I'm not really a candidate for Paxlovid. She only gives it to high-risk patients. Well, according to Daniel, that's what the EUA is for, right? People at risk. It's not supposed to be given to a 25-year-old, healthy 25-year-old at the first positive test. That's not what the EUA is for. Daniel says that all the time, so that's correct. The vaccines aren't updated because supposedly they still work, but actually their effectiveness is lower because of immune escape. Why wouldn't a new updated vaccine work better? Well, I'm not sure that their effectiveness is lower because of immune escape, okay? I'm not sure that the metric being used, which is hospitalization, is the correct metric, all right? That's just, I don't want to see hospitalization because that varies depending on what hospital you're in. And if you make a new vaccine, then more variants will arise. I'm not sure that's any better than the one we have. Any thoughts, Amy?
You can take it. That's fine.
Okay. If given the opportunity to study the virus at home, what is the first experiment you would perform in your garage? Which virus?
Plaque assay.
SARS-CoV-2?
Any virus. Plaque assay.
Well, that's not an experiment. That's a technique. So what would the question
What are
be?
you talking about? Does it plaque as an experiment? It's yes or no. Plaque or no?
I would like to see how long a person who's vaccinated and infected sheds infectious virus. That's what I would do.
I wouldn't do that.
Well, we all would do different things. There's a paper, 830 million-year-old microorganisms in primary fluid inclusions in halite in the journal Geology. Microorganisms. Could virus be found too? You might find viral genomes, not infectious
Depends on the virus.
DNA
How do you know?
viruses, yeah.
You're just, I was going to say, you can't say a DNA virus could not be found. You might argue that an RNA virus cannot be found, but a DNA virus is probably very stable.
Yeah. I mean, the DNA, right? I
I
think
don't know. You can
infectivity
have,
would
I
be
don't
there.
know. How stable is the adenocapsid? Non-enveloped proteinaceous particle.
What's the oldest DNA virus recovered? 30,000 years from permafrost. So it was frozen, right? One of those giant viruses. So I'm not sure if it's not frozen, it would last a long time. Are you going to retire from Columbia anytime soon? No, I'm not. I'm still going to be there. I'm going to teach and be an expert source of information. But I do spend a lot of time at the incubator. And that's the other thing. That's what I want to do. I want to produce science educational programs with your support. So the wall of polio doesn't have to be moved right now. We're just thinking ahead, right? This is funny. Make the wall of polio freestanding and on rollers. That's good.
Yeah, and so what is going to roller skate around Rockefeller Center?
Yeah, first commercial Hep B vaccine was 41 years ago. I guess I was wondering why my NP asked about it last month. Yeah, just get another shot. Just get another shot. Considering one could replicate Foucher's experiment, how many generations of animals would you suggest? Oh, the adaptation of H5N1 to droplet transmission? I don't know what you mean by generations. He did 10 passages, I think. Something. But ferrets are rather expensive. I wouldn't do that in my garage. No. Wouldn't do that.
Dirty animals. I don't want any.
Predict.
No dirt.
Schmicked. Amy is awesome. It's getting darker there, Amy.
Yeah,
HIV
it's nighttime. That's
positive
what happens at 10:15.
41-year-old developed cell clumps in various organs. Sequencing results 99% tapeworm, not human cancer. Yeah, I thought we did something similar. One of Daniel's clinical cases was something like a cancer, a colon cancer with a worm in it or something like that. I'll take a look. Thanks. Thanks. How do cell tropisms affect viral infections? Amy, do you want to take that?
They limit it and they reflect the disease that viruses infections can result, right?
Yeah, I mean, depending on where the virus is reproducing, yeah, you can have different disease outcomes for sure.
Mm-hmm.
Any opinion on the Chinese government commitment to maintaining a naive population? It's silly, not maintainable.
I didn't think I mean, I don't think that that's their primary goal. I think that's a secondary consequence for excuse me, a not good public health policy because their population is too big.
Looking at ventilator numbers when determining if things are getting better, I think a ventilator number is much better than just hospitalization, because many of the hospitalizations are not even for COVID, they're with COVID. So that
I
really
don't
isn't
think a
fair.
ventilator number is good.
You don't think so?
No, because many people aren't on ventilators.
Well, I think death is the best metric, and then it's so low now in a vaccinated population that it's hard to get good numbers.
Well, that's what you want, so I think it's fine.
Yeah, I agree. Can you say something about Peyer's patches in the intestine and how they function? Peyer's patches are collections of lymphoid tissues. And the lymphoid tissues, for example, where antigen-presenting cells go, they pick up antigens from the periphery, and the gut would be the gut lumen, and they present them to T cells in the Peyer's patches, and the T cells decide if it's foreign. If it is, they get activated and start an immune response. So kind of think of them as the local lymph nodes in the intestine. Is that fair, Amy?
Yeah, for sure.
Would Amy's Columbia lab be reassigned to someone else, or is it closed for good? I don't know.
It's been reassigned to somebody else.
Someone's getting half of it. So we never had more than half of the lab, and so the half we didn't have is going to be reassigned. The half we used is still ours for now, and it's got stuff that Amy wants to ship out. Right, Amy?
Yeah.
Okie dokie, we're getting to the end. How is a specific strain variant isolate of a lineage isolated in the lab just to study that specific change? So if you get a isolate from a patient, let's say SARS-CoV-2, your isolate's going to be different from my isolate, so they're both variants. And so people can take those and study them in the laboratory. A strain is something that has a very different biological property, and that would have to be determined by various experiments. And so if you want to know if a certain change has an effect on a virus, then you introduce that change on its own into the virus and then study it. Gribbler says the divergence from monkeypox is the number of variations divided by the total length. That could be. Does that sound good, Amy?
Yeah, sounds right. Sure. Sounds good.
Amy, did you say what your new title will be? I'm at the FDA CBER website looking for you.
I'm not on it yet. I'm not an official member yet. It's cute.
Do you have a title yet?
No. Just Amy Rosenfeld, PhD.
Well, you're going to have a title once you're officially hired, right?
Sure.
What's the average time from
semantics? Am I really all that concerned about it?
Well, they're interested, Amy. They're asking you a question. They'd like to know the answer. Whether
I know,
you care or
not
is a different issue, right?
Well, that's why I don't have an answer, is because I'm not concerned about that answer, that question. I'm concerned about where my viruses are.
What's the average time from exposure to symptom onset for current variants? Tired, Amy?
Well, since symptom onset is the reporting of the immune response, it could be 2 days.
Yeah, it can be 2 to 3 days. That's what Daniel is
But
saying.
it's not the symptoms caused by the virus. It's the symptoms that's caused by you having a
Yeah.
primed immune response. So it's not correctly interpreted. It's unfortunate that a certain person who was at a prestigious university and now at a company is screwing up.
So excited to have you in our FDA.
Thank you. I'm excited to be in the FDA.
Amy, are you planning on retiring early? Amy will never retire. You heard it here. You heard it here.
This 2 weeks is driving me crazy.
What's the best bagel place in Midtown?
Midtown?
Yeah.
Some people like Essa bagels, but they're kind of big and doughy. They're on like 53rd and Lex. There's Bagel Talk on Amsterdam at like 77th that we sent Petra to. There's not a lot of bagel places in Midtown that I can think of.
Maybe you could tell us what branch you will work in. It's CBER, right?
Yeah, the Center for Biological Evaluations and Review. Yeah.
So Andrea is now pumping out all these texts from this paper, Andrea. This paper is flawed. We're going to actually do it on TWIV on Friday. It's in a older population. It's not the general population. It's veteran affairs population. And I just find it hard to believe that there's no difference in long COVID in vaccinated versus unvaccinated people after an infection. I just don't believe that. It doesn't make any sense to me. I think the diagnosis for long COVID is flawed, and I think the population is flawed. So just take it with a grain of salt. H&H bagels?
I don't like them.
How about Essa?
That's the one I just said. That's on like 53rd and Lex. But they're kind of like the ones that you get at the Bagel Pub. They're very large and doughy.
It seems like my hamster farm has become resistant to SARS-CoV-2. How do you know this? What sequence do you want to share? Your hamster's sequence?
I'm not even sure that all hamsters are susceptible to SARS-CoV-2, or only Syrian golden hamsters, which is a very specific kind of hamster. So I don't know. Maybe he has the non-susceptible home version of the hamster. I don't know. I don't know enough about hamsters.
Well, we've reached the end of the questions at 8 minutes before the hour, so I think we can wrap this up. I want to thank everyone for coming. We have over 500 people. That's awesome. Thanks everybody. Thanks for your likes. Maybe we could get the likes over 400. That would be great.
Well, everybody should push the button until their finger falls off. It's not like you only push it once. Just push, push, push. Be like the kid in the elevator who pushes every button. So the elevator stops at every floor. Drives me bananas.
I want to thank the mods tonight, Tom, Les, Steph, and Vanity Nutrition for help keeping things civil. Thanks all of you for coming tonight with your questions. I really appreciate this lovely community that we have made. And Dr. Rosenfeld stayed to the very end. Everyone should thank Amy for that. And I suppose that since you're doing these at home, if you can continue them, you'll always stay to the very end, right, Amy?
I guess so. We'll
unless
see.
You have
we'll to,
unless you have to run back to the lab late at night to do a plaque assay, right?
Time points, you can never tell. I've been known to go to the lab at 2 a.m. I used to walk through the snow in the middle of Montreal. It was not so much fun.
Thank you, Amy.
All right, thank you. I talk to you tomorrow. What are we doing tomorrow?
I'm working
Oh,
at
we're
the
looking at the
incubator.
article. We're looking at the
What
article
article? Is it
tomorrow.
done? Is it done?
No, needs
Okay.
more work.
Thanks everybody
It's
for
getting
coming.
done.
We will be back next Wednesday at 8 p.m. Good night, everybody.
Yes,
Thank you.
good.
Bye-bye.
Bye.